Hi everyone. Thank you for joining me today. My name is Justine, and it is my pleasure to give this presentation today on invasive earthworms and their effects on soil carbon in the boreal forest. As many of you already know, the boreal forest is the largest terrestrial reservoir of carbon, and this important biome is being invaded by European species of earthworms. The invasion of earthworms in North America has started with temperate forests, and they're now reaching the boreal forest. So most of the studies have been done on temperate forest. So there is a need to research that topic in boreal forests. Based on this background, we have three axes of research. We are wondering if we can expect to see similar effects of invasive earthworms as what has been recorded for temperate forests. We are even more so interested in effects on the mineral soil because the little information we have is focused on the forest floor. And because we know that earthworms are basically feeding on organic matter and incorporating that organic matter with mineral soil, there is a huge bioturbation action, and there is a high chance that this action has an impact on soil carbon dynamics. We have selected a total of 7 sites across the Canadian boreal forest. 3 of those sites are located in Alberta, in the boreal plains with alluviosolic soils and an aspen-dominated canopy. 4 of the sites we have selected are located in Quebec, in the boreal shield, and we have 2 sites with pozolic soils and a mixed conifer canopy, and 2 sites with brunisolic soils and maple tree canopy. Our experimental design is based on a space for time approach, meaning that we made the assumption that our sites invaded by earthworms were similar to our control sites before invasion. So when selecting sampling points, we made sure to have similar canopy cover, similar parent material, similar slope, and similar aspect. And to further validate our approach, we ran preliminary analysis, including pH and texture. As I mentioned before, we studied 3 different soil types, so brunisols, pozols, and alluvisols, that are representative of the upland boreal forest. For 5 out of 7 sites, we were able to have a site-level paired design. This means that we could find within a site a zone that was invaded by earthworms and a zone that wasn't, that was used as a control. The first step of our approach was the description of soil profiles with and without earthworms. These 2 photos are a good example of what we found in most cases. This is from a site close to Quebec City on the military base of Valcartier with brunissols and maple tree canopy. These 2 pedants were less than 100 meters apart, and the only difference we could find between them was the presence of earthworms in the pedant on the right. In this invaded zone, the forest floor fully disappeared into a mole, and we can only see a few discolored leaves on the surface and many earthworm casts. For the mineral soil, we also found differences in structure. It was also enriched in organic matter compared to the control. To summarize that descriptive part of our research, we observed that the forest floor was greatly reduced following invasion and even disappeared in extreme cases. The structure of that forest floor also changed. There was a loss of the humic, so the most decomposed horizon, and an overall simplification of the structure. We found many earthworm casts in the invaded forest floor, and according to the classification for forest floor by Klinka, we could classify the forest floors as vermimole in all cases, regardless of soil type. We also observed the incorporation of mineral particles, which is a clear sign of bioturbation. And for the mineral soil, the structure of the top mineral horizon was changed. It was enriched in organic matter, mainly made of earthworm casts. So we classified it as an AHU, H for organic matter, and U for bioturbation, which is, by the way, part of the Canadian system for soil classification, but that U suffix is not very often used. And those observations are valid for all soil types that we studied. To complement this descriptive approach, we also estimated carbon stocks for the forest floor and the surface mineral soil. We decided not to sample further down in the profile because that's not where we were expecting earthworms to have the greatest impact. So on this graph, you can see the three soil types that we're studying. So brunysols on the top, then podzol, and then luvisols. On the left, it's control. On the right, it's invaded. The darker the color, the more carbon there is. And significant differences between invaded and non-invaded are displayed with the p-value or ns when not significant. So what is clear here is that there is a huge loss of forest floor in brunysols, 59%, and even more so in luvisols with 94%. For the mineral soil, there is in most cases no difference, and there is not enough of a difference to see an actual trend. What is clear though is that the decrease we observe in the forest floor is not compensated by an increase in the mineral. And the degree of effects is the highest for luvisols and the lowest for podzols. We have seen that the carbon found in the forest floor is greatly reduced and that in the mineral, there is no clear trend. But even if there is no clear trend for the total carbon that is present in the mineral, the A horizon was reworked by earthworms. The structure is different, the density is different, and not all carbon is equal for decomposition and stabilization. So the question raised by these findings is the link to persistence of the carbon left in the forest floor and the carbon transferred in the mineral. Is it going to be more active, more stabilized? And to answer that question, we set up a laboratory incubation at constant temperature in the dark for 1 year. We homogenized field replicates to create 4 composite laboratory replicates for both invaded and non-invaded soils. I just want here to say that we did not incubate soils with earthworms in the lab. We just used soils that had been affected or not by earthworms. We periodically measured accumulated CO2 with the Picaro, and we considered that the cumulative CO2 emitted over 3 months was considered fast carbon, and the total over the entire year would be active carbon. And the results are very clear and very interesting, in my opinion, in the sense that the forest floor is not greatly impacted. Although we lost a lot of carbon, the carbon that's left seems to have the same activity or persistent as before. But for the mineral soil, after invasion, as you can see circled in red, the proportion of active carbon is greatly increased. So the graph is looking at the proportion, the percentage of active carbon, not the total amount of carbon. But the trend is the same regardless of soil type. So no clear trend for active carbon in the forest floor, but a very clear increase for the mineral soil. The question now is what is behind the increase of active carbon that we observed after earthworm invasion? And we are currently investigating 3 potential mechanisms that could play a role in the results that we observed. The first mechanism is the physical protection of carbon within aggregates. Because of earthworm activity, bioturbation, and cast formation, there might be an impact on the physical availability of carbon. And we are approaching this by analyzing the total carbon found in different soil fractions. The second mechanism could be the chemical composition of soil organic matter. We know that earthworms feed on specific compounds in the soil. They usually prefer leaves over roots. So we are investigating the overall composition of organic matter by pyrolysis, gas chromatography, mass spectrometry. And we're looking at aliphatic and aromatic compounds. We're looking at cutin and suberin biomarkers. We're looking at carbohydrates. And we're hoping to see maybe a difference in the overall composition, maybe a shift. The third mechanism is regarding microbial activity and microbial communities, because in the end, microbes are responsible for carbon mineralization. And we are trying to characterize microbes using two different methods, phospholipid fatty acid, PLFA, for a more quantitative approach, and DNA for a finer scale resolution. The preliminary results we have for PLFA showed that the ratio of gram-positive to gram-negative bacteria is lower in invaded soils. And that would be definitely linked to the increase in carbon availability that we found from our lab incubation. So I'm hoping that I will be able to present more results in the next few months or so. I would like to thank you for your attention, and I will be happy to answer any questions now, or if you want to contact me later, I just added my email address there. Thank you.